So welcome, everybody. Welcome to this year's 5th Anne Klibanski Visiting Lecture Series. My name is Jody Gilman. I'm the Director of Graduate Student and Postdoctoral Division in the Center for Faculty Development. And today, our own Rebecca Gillani will talk about neuroinflammation causes neuronal dysfunction by destabilizing excitatory synapses. And Dr. Maria Hastermann from the Max de Planck Center in Berlin will present on antigen-specific T cells in autoimmune inflammatory diseases of the central nervous system. Thank you so much for being here today. The two speakers today, Drs. Gillani and Hastermann, have disclosed that they have no relevant financial relationship that conflict with today's lecture. Okay. So before our speakers begin, I want to share a very brief history of the Anne Klibanski Visiting Scholar Award Program. It was created by the former CFD director, Miriam Burdella, to offer MGH women faculty an opportunity to serve as a virtual visiting professor and give grand rounds or other special lectures at a national or international institution organized by the Scholars Champion, a senior faculty member at MGH and the CFD. And this is Anne Klibanski. She was the president and CEO of Mass General Brigham and the former director of the Center for Faculty Development. A highly successful researcher, Dr. Klibanski has been a strong supporter and advocate for women throughout her career. We've had 4 cohorts of Anne Klibanski Scholars to date. Here's the inaugural cohort of 36 scholars. And here is the second cohort in 2021 to 2022 with 32 scholars. And the third cohort, 2022 to 2023, had 31 scholars. And we have 21 scholars in this year's 4th cohort. Today's MGH speaker, Dr. Gillani, is from this magnificent group. This program would not be successful without the commitment and energy of our Anne Klibanski Scholar Champions, who are helping this year's scholars find exciting speaking engagements. And again, the goal of the Anne Klibanski Visiting Lecture Series is to catalyze and foster collaborations across national and international institutions. And here are some of the institutions we've collaborated with through the years, and highlighted in yellow are the Max Delbrück Center in Berlin to represent Dr. Hastermann. And now, without further ado, I would like to introduce our speakers. So first we have Dr. Rebecca Gillani, a clinical expert in multiple sclerosis, a member of the Mass General Neuroimmunology and Neuroinfectious Diseases Division and the Mass General Multiple Sclerosis Clinic, an instructor in neurology at Harvard Medical School. She's a neuroscientist and principal investigator in the Mass General Institute for Neurodegenerative Disease. Her research focuses on inflammation-mediated neuronal dysfunction in models of multiple sclerosis. And here is our second speaker, Dr. Maria Hastermann, a principal investigator and physician at the Experimental and Clinical Research Center in Berlin. I'm not going to try to pronounce that in German, but thank you so much for being here with us. She completed her postdoctoral training at the Institute for Integrative Neuroanatomy in the Neuromuscular Group from 2016 to 2018. She was a visiting scholar at the Clinical Neuroimmunology at the NeuroCure Clinical Research Center. So, Dr. Gillani, I'm going to pass the virtual microphone to you.
Okay, thanks everyone for joining today. I'm really excited to be able to talk about some work that we did in my laboratory over the last few years. And as mentioned before, I don't have any conflicts of interest to disclose today. So first, I want to tell you a little bit about multiple sclerosis. So this is an autoimmune inflammatory disease affecting the brain and the spinal cord. And people with multiple sclerosis develop demyelinating lesions in the brain and the spinal cord. And here we have an image from a brain MRI from a person living with multiple sclerosis, and we can see examples of these demyelinating lesions within the brain. And depending on the location of where these lesions develop, they can lead to relapses of neurological symptoms. And it might be natural to think that most disability accumulation in people with MS might be related to these relapses. But actually, in the last few years, we've learned that that isn't the case. So in this study, what you can see is they looked at total confirmed disability accumulation in this clinical study of patients being treated with a medication called ocrelizumab. And then they broke it down to determine how much of this disability accumulation was related to relapses and how much was independent from relapses. And what you can see is actually in the orange, only a small minority of disability accumulation is related to relapses. The majority of worsening is actually independent of relapses and was termed PIRA, or progression independent of relapse activity. And you can see in this particular study, this was from two randomized control phase 3 trials where they compared ocrelizumab to a more modestly effective medication called interferon beta 1A. What they found is that while this high efficacy medication did decrease the risk of confirmed disability accumulation and PIRA, a portion of these patients did continue to develop confirmed disability accumulation despite treatment with high efficacy multiple sclerosis disease modifying therapy. So the question is, what is underlying this worsening in neurologic functioning despite controlling relapses? And the answer is that a main contributor is likely neurodegeneration. So previously, neurodegeneration was primarily thought to be a late process in multiple sclerosis. But more recently, we've realized that that actually wasn't true. Neurodegeneration actually begins at the earliest time points in multiple sclerosis, and it actually occurs the fastest at earlier time points. So in this representation, this figure, you can see that they're showing us that cortical gray matter volume loss is more accelerated in younger people with multiple sclerosis, as is retinal atrophy, which is accelerated in younger people with multiple sclerosis. So what this tells us is that neurodegeneration is an important early pathology in people living with multiple sclerosis. But what is the pathology that underlies that neurodegeneration? Well, a really important component is the loss of excitatory synapses, which has been shown in multiple studies. We're just going to look at a panel from this one particular study here. And what these investigators did is that they took postmortem samples from people who had longstanding multiple sclerosis and then looked at pyramidal neurons located in the cortex. And you can see this is a dendritic segment from a healthy or a control that did not have neurological disease. And what you can see is that in the samples from people who had multiple sclerosis, both within demyelinating lesions and also within the normal appearing gray matter, there was dramatic loss of dendritic spines. And dendritic spines are the anatomical location of excitatory synapses. So what this translates to is that people with multiple sclerosis have the massive loss of excitatory synapses. Now, of course, when we're looking at a postmortem study, we're looking at one point in time, and typically these come from these studies include patients who had very long-standing multiple sclerosis. Now, what we wanted to ask the question was, how does neuroinflammation shape neurons and synapses very early on as they are first being exposed to neuroinflammation? And of course, in the past, we thought of as synapses as fixed structures, but actually that's not the case at all. We now know that they're very dynamic structures, changing in response response to the environment and as the organism learns and forms memories. So another question we wanted to ask was, how are synapses dynamically altered by neuroinflammation? Not just looking at one time point, but over time. So to answer these questions, we used a model called experimental autoimmune encephalomyelitis. And this is a model that causes neuroinflammation within the brain and the spinal cord. And the way this works is that we inject a peptide sequence that is found in myelin into mice, along with inflammatory adjuvants. And this immunization against this myelin protein causes a autoimmune neuroinflammatory response within the brain and the spinal cord. And that spinal cord inflammation and demyelination leads to clinical signs of ascending paralysis in the mouse. But we also took advantage of the fact that these mice also get inflammation within the cortex of the brain, including with small demyelinating lesions. So here in these experiments, we're asking the question about how does this neuroinflammation, this cortical neuroinflammation, shape neurons and synapses? So in order to do that, we needed to label the neurons and their synapses. So we used a technique called in utero electroporation to introduce plasmids into newborn neurons in fetal mice. And when these mice are born and grow to adulthood, those neurons express YFP, a fluorescent protein, as cell fill. So it fills the neurons and their dendritic tree. And they also express a fusion protein of a postsynaptic protein called PST95 bound to a fluorescent protein called teal fluorescent protein. And so what you end up with is that these mice have fluorescent labeling of the neurons as well as their excitatory synapses. And then when these mice grow to young adulthood, we implant a cranial window to get optical access to the brain. And then we induce the mice for EAE and then use multiphoton imaging to obtain images of these neurons, individual neurons, in the somatosensory cortex longitudinally over time as neuroinflammation develops within the brain. And here's just a little cartoon to show what this looks like with our neuron with its complement of dendritic spines and labeled excitatory synapses. And here's an image from our 2-photon microscope. This was obtained in a living mouse through its cranial window. And what you can see here is that the YFP is pseudocolored in red. So you can see the cell body and the dendritic arbor of this neuron. And you can also see its complement of dendritic spines and excitatory synapses. And what we're going to do is we're going to zoom in on this short segment. And then these are images of the same segment that were obtained at weekly intervals. And what we can now see is that this technique allows us to follow the fate of individual dendritic spines and their excitatory synapses over time. So the first thing we looked at was the turnover of the dendritic spines. So as expected, in the pink, you can see that in a healthy brain, there is a low level of normal turnover of dendritic spines over time. But this is accelerated in the setting of neuroinflammation. And what's really interesting is this actually starts even before the onset of the ascending paralysis symptoms. So here we're giving clinical scores to the mouse of the paralysis, and you can see that the mice started to exhibit symptoms around day 12, and yet we were able to detect this increased instability of dendritic spines even at this early time point, comparing 0 to 7 days after immunization. Now, the gains and the losses of these dendritic spines was actually balanced. So what that meant is that despite the fact that these dendritic spines have increased instability, we're actually not losing dendritic spines over time. But you might be wondering, well, you labeled that excitatory postsynaptic PST95, that excitatory postsynaptic protein. So what happened to the synapse? So we were expecting to see loss of PST95. So basically the idea that the loss of the excitatory synapse within the dendritic spine may contribute to that instability. But actually we didn't see that. So we saw that despite the fact that these dendritic spines have increased instability, they still retained PST95. Well, now you might be thinking, what about the presynaptic side? So here we're looking just at the postsynaptic protein. Could it be that the instability is explained by the fact that they are losing their presynaptic partner? So to answer that question, we needed to use a different technique. So we used a tissue expansion and clearing technique called magnified analysis of the proteome, or MAP. And what we did is we took tissue samples, brain samples from mice at the peak of EAE severity. So a clinical score of about 3 or a 3.5. And then we processed this tissue for MAP. And this is actually what a dendritic segment looks like when you look at it at super resolution. So you can see now that these dendritic spines, you can see the fine detail of these dendritic spines and their spine heads. And we're going to zoom in on 3 different dendritic spine heads. And when we do that, we can see here that we labeled for PSD95, that postsynaptic scaffolding protein. And we also labeled for bassoon, a presynaptic scaffolding protein. And this is from an EAE brain. And what we can see is that actually qualitatively, synapses look pretty normal in the setting of EAE. We have that normal apposition of PSD95 and then the presynaptic bassoon. So here we have like a horseshoe-shaped synapse, a punctate one, and then a perforated, which are all sort of normal shapes for synapses. Now, when we quantified this, we found that indeed the majority of dendritic protrusions in the mice with EAE did retain presynaptic bassoon and postsynaptic PSD95. However, a small minority actually did lose their excitatory synapse, either through losing both PSD95 and bassoon, or just one or the other. So these dendritic spines and excitatory synapses are unstable in the setting of EAE, but only at this time, at this early time of inflammation, only a small number of actual excitatory synapses are being lost. Now, you may recall that we looked at the same individual neuron longitudinally over time. And we did that because we wanted to ask the question, how does the excitatory input that an individual neuron sees change over time when it's being shaped by neuroinflammation? So the first thing we did is we classified the dynamic behavior of individual dendritic spines. So basically, what was their fate over time? And we classified these for the sessions done at 7, 14, 21, and 28 days after immunization. And we classified them as stable, so the dendritic spine never changes. One-time dynamic, meaning that a spine is formed during the imaging session or is lost during the imaging session. Transient, it appears and then is lost. Or recurrent, it's coming and going at the same location. And what we saw is that, as expected in the setting of EAE, the proportion of stable dendritic spines drops. And contrary to that, the number of one-time dynamic dendritic spines increases, as well as these recurrent dendritic spines, which are actually pretty rare in the healthy animal. So the bottom line for this is that despite the fact that there is this instability of these dendritic spines, these neurons do retain the capacity to form new excitatory connections. And then we wanted to ask, how are these changes distributed across the dendritic tree? And to do that, we reconstructed these neurons using a program called Neurolucidus 360, and then placed markers across the dendritic tree for these different dendritic spines. And we classified them as either stable, so never changing, or dynamic. So any of those categories of dynamic that I showed you before, any dendritic spine that changed during the imaging. And you can see for this individual neuron, here's the reconstruction with, these are the segments that were scored for this particular neuron. And you can see in green, we have the stable, and magenta, we have the dynamic. So first we analyzed this doing a nearest neighbor analysis. And what we saw is that on average, a dynamic dendritic spine in the setting of EAE will be closer to the nearest other dynamic dendritic spine. So you can actually see that this is a pretty significant change, and that the distance between the dynamic dendritic spines is cut in half in the setting of neuroinflammation. We also noticed that when you look at stable synapses and look for their nearest neighbor, dynamic neighbor, you can see that stable synapses are also in closer proximity to their nearest dynamic neighbor. And this is relevant because, when we think about nearby synapses influencing each other, generally, thinking of synapses maybe in the region of about 10 microns of each other can influence each other. So by these becoming closer to one another, this can influence the nearby synapses. Then we asked about how are these distributed across the dendritic tree? Are these dynamic events really focused in one particular area, or is this a diffuse process that happens throughout the neuron? And what we saw is actually that these dynamic dendritic spines in the setting of EAE occur across the dendritic tree. So here we're showing you the density of these different types of dendritic spines from distance from the cell body. And in purple, we have those dynamic dendritic spines, and you can see that they're increased across the entire dendritic arbor. And here we quantify and show that as expected, the density of these dynamic dendritic spines is increased in the setting of EAE. But it doesn't seem that they're focused to one area. It actually seems that they follow a similar pattern that we see in the healthy neurons, by which dendritic spines typically tend to be lower in density closer to the cell body and then increase in density as we get further from the cell body. And it seems that those dynamic dendritic spines developed in that overall normal pattern. And to further look at that, we made bins starting at the cell body for this dendritic, the dendritic spine density, starting with 0 to 49 microns from the cell body, then 50 to 99 and 100 to 149. And then again, this demonstrates to us that that this does appear that the dynamic dendritic spines are increasing across the dendritic arbor, following a relatively normal pattern where there's lower density near to the cell body and higher as you get further away from the cell body. So today I've shown you that neuroinflammation has destabilizing effects on excitatory synapses. But despite the fact that there is this instability of excitatory synapses in EAE, the neuron retains the capacity to form normal appearing excitatory synapses and does retain the ability to form new excitatory conditions, which I think represents an opportunity, and that if early in the setting of neuroinflammation, we can establish the right conditions, we may be able to allow neurons to form more normal excitatory connections. And then future direction. So of course, it's really important to understand what are the mechanisms driving this instability and how can we target that therapeutically in a person who's being affected by neuroinflammation. So different pathways that have been implicated are soluble pro-inflammatory molecules. And so this may explain the changes we see in synaptic dynamics even before immune cells migrate into the brain in EAE. It could be that these inflammatory molecules are making it to the brain early. So those include complement pathways, interleukin-1 beta, and tumor necrosis factor alpha. It's likely that also cells in the brain contribute to this synaptic pathology, including neurotoxic astrocytes and microglia, which may engulf synapses, as well as mitochondrial dysfunction. So we know in the setting of neuroinflammation, neurons have mitochondrial dysfunction, and neurotransmission is a very energy-intensive process. So this mitochondrial dysfunction can contribute to synaptic dysfunction. Thank you, everyone. From the Mass Journal, I have a list of people here who have contributed to this work in some way. Thank you to our funders, and also thank you to Professor Ellie Nadivi at the Massachusetts Institute of Technology, who kindly gifted the plasmids we used in this experiments, as well as others at the MIT who gave advice on these experiments. Thank you for your attention today.
Is it okay to ask questions?
Yeah, go for it.
Okay, fantastic. Well, Becky, that was, a fantastic body of work, and congratulations on your new publication. Very well deserved. The question I have is, in the context of MS, right, what is the significance of synapses, coming and going? So when we think about, synapses being lost, right, that would account for the loss of function, whatever that may be in the brains of these mice, and, ultimately translating to the loss of function, cognitive function in patients. So what would be the functional significance of synapses appearing, new synapses appearing and then disappearing, as you showed in some of your dynamic synapses?
Yeah, I think one way that you can think about this is particularly with regards to cognition, as you mentioned. So when you think about the dynamics of dendritic spine, so this is actually not random in the healthy brain. There's some really elegant experiments that have been done recently that show that what happens in the setting of learning and memory is that new dendritic spines are targeted to clusters of coactive dendritic spines. So you really need to be able to target that new excitatory synapse to a particular location on the neuron to serve learning and memory. So I think in neuroinflammation, where everything's destabilized, I think that sort of explains how you can have cognitive dysfunction, because you're just not able to target those new synapses to where they need to be to serve learning and memory. And that's very relevant in people with multiple sclerosis, because the majority of people with MS actually struggle with cognitive dysfunction greater than 50%. And it may also contribute to the profound fatigue that some people with MS suffer from as well. So I think it is a very, like you said, I think it's clinically relevant for progression, but also very clinically relevant for cognition, as you mentioned.
Great, thank you so much.
Are there any other questions?
I had a question. Great talk, Becky. That was fantastic. And along those lines, thinking about our MS patients, in addition to getting a control of the inflammation, could you think of like a therapeutic along the way that maybe in addition, kind of helping to compensate by boosting like electrical stimulation, maybe potentiating AMPA receptors or something? Is there something else that could potentially help compensate in addition to kind of getting control of the inflammation that's driving this process?
Yeah, I think so I think there's like two things I would think about. One would be, I showed this long list of pathways that may be causing neuronal dysfunction and synaptic loss. So it may be that we need to think a little bit differently than our current approach in multiple sclerosis, where we typically only have patients on one disease-modifying therapy at a time, as opposed to, rheumatologic conditions where someone may be on multiple immunosuppressive medications at the same time. So could it be that potentially using a treatment that targets, for example, inflammatory cytokines or astrocytes or microglia in combination with another disease-modifying therapy may be neuroprotective? I think so. And the other part of it would be better understanding this mitochondrial dysfunction that occurs in neurons in the setting of multiple sclerosis. So perhaps protecting neurons from mitochondrial dysfunction would be neuroprotective as well. So I think, yeah, I do agree. I think we'll have to change the way we think about treating people with MS, where we may need to be using multiple different medications to treat other different aspects of the pathology.
Yeah, and for example, like remyelination is also really energy dependent as well. So I totally agree, like mitochondrial dysfunction could be a very nice avenue to start to approach. Great.
there are no other questions, should we start our second half of the talk?
Thank you, and hi from Berlin. It's a great opportunity to talk and present my projects with you guys. And I'm very happy also to see Becky again. So we have met last year during a MOGAT meeting. Yeah, and hopefully we will go on having some nice cooperation with this fascinating work she's been doing also for our topic. So I'm very looking forward to that. So yeah, I was already nicely introduced. So I'm in Berlin currently. I think I can also show you just a little overview. So we have several sites in Berlin where we are conducting research as well as patient care. So I hope you can see my mouse. So I'm currently here sitting in the labs, but also we have here our outpatient clinic, which is led by Judith Bellmann-Strobel, who is also doing patient care as well as several studies, including her specific point of looking at fatigue, not only in multiple sclerosis, but also like post-COVID and other conditions. And then our other main site is in Mitte, Charité Mitte, where Tanja Schmitz-Hübsch is leading also many other studies that are investigator-driven and also pharma studies regarding all kinds of conditions from patients, including cognitive and motoric analysis and so on. So just to give you an overview, what is basically being done in Berlin. So we are led by Professor Friedemann Paul, and he has set up this Berlin registry cohort where we have the target to collect around 650 patients, primarily with MS, but also other rarer conditions like neuromyelitis optica and MOG-associated disease, which are both more like my main topics, as well as other isolated symptoms. And the idea is to actually follow those patients for a long period of time, like around 15 years, in yearly visits, and also include healthy controls. And the patient have quite the program when they visit us during that day. So we are trying to really have a very broad spectrum covered of analysis that we are doing with them, including imaging, MRI, OCT, a lot of questionnaires that you can see here, metabolic analysis, as well as biosampling. So we try to have a real good biobank of all these patients. Yeah. And furthermore, Professor Paul also has set up the NEMOS network with some other people. So at the moment, there are over 25 centers in all of Germany that are participating to also collect samples from these rarer diseases like neuromyelitis optica and MOG-associated disease, so that we can share the data as well as biosamples from these patients and get a better insight into these conditions. So this is my team in Professor Paul's lab, my 3 PhD students, as well as 2 medical students, and some of the cooperation partners, but of course there are many more. And so I will just jump into the topic. So as Becky has been telling you, a lot of the disease, the EIE, she was inducing with a MOG peptide, which is a myelin peptide. So here you can just see a little of the structure. So it's located on the outside of the myelin sheath, and is actually rather low abundant. And while the MOG protein has been used since, I think, the 1980s as a model for multiple sclerosis, that you can see here. There have been developed a lot of different models to look at different aspects of multiple sclerosis, but also other diseases. And so just that there's no confusion later, I wanted to just divide the different main types of EAE. So probably you know it already. I will just say 2 words about it. So Becky was using the active EAE, where basically it works like a vaccination. So active immunization, meaning you have an antigen of interest that is injected together with the adjuvant to induce the disease in the animals, which takes about 10 to 12 days. And then there are different disease courses depending on the animal. And then the second approach that we have been using a lot is a passive EAE induction, where after immunizing the animal with an antigen of interest, you would isolate the lymph nodes, the inguinal lymph nodes, and propagate the T cells in vitro culture until you have expanded them to sufficient numbers to perform an adoptive transfer into the animal, where you actually know which T cell antigen specificity you then have. And here you can see just an overview where they have compared, where they show basically where the main lesion locations are in multiple sclerosis compared to mostly mock-induced EAE models. And here you can see that just by having different lines of mice and rat, you already have quite the different lesion locations. And furthermore, it has been shown also that like last year, that actually it seems that patients that have autoantibodies against myelin oligodendrocyte glycoprotein probably a separate disease entity as well to multiple sclerosis. So here you can just see, I mean, in the clinic, they can look very similar to multiple sclerosis. You can see here the inflammation in the optic nerve, which is very characteristic, as well as the inflammation in the spinal cord. It is a rare disease. And it can be either monophasic or have a relapsing disease course, and it has the specific autoantibody. However, this is a bit of a problematic situation, because also multiple sclerosis patients can have their autoantibody, although there it is at a lower concentration. And here you can just see some other images from our cohort, where you can also have longitudinal lesions or just spots, as well as a very characteristic age sign here in the gray matter. And so to get to the field that is interesting to us, you can see here that the lesions are actually infiltrated quite a lot by T cells. Also B cells are present. So these are 2 studies, one from Europe and one from Japan, which are looking actually quite similar in this regard. And you can see here that there are predominantly CD4 T cell mediated infiltrates here. So what has been interesting to us is that with different antigen specificities, you have different lesion locations and also the different formation of lesions. So we also have performed a passive transfer EAE with MEP as specific, MOC specific, and S100 beta. It's a astrocyte protein specific cells, and you can see here that the amount of cells entering into the CNS is quite different, with MVP being quite low. S100 beta, a lot of T cells enter, and MOC, just some cells enter. And we have analyzed those cells that were entering and could see that the reactivation profile of these cells was quite different between the different antigen specificities, hinting that also the reactivation in the brain is quite relevant to the lesion formation forming later. And also you can see here that you have a different amount of interferon gamma being expressed by the cells with the different antigen specificities here. And the interferon gamma, as also Becky has hinted, here has a lot of implications, for instance, on microglia and their production of complement factors, and then the consecutive formation of lesions. So, oops, sorry. So here again, also you can see different like older studies that already have also looked at the location of the lesion in the spinal cord and the animals. I can see here subpile distribution here with mock white matter distribution and the MBP, a quite abundant distribution of lesions, and astrocytic protein also like more white matter lesions. And this is actually quite interesting because as you can see here, the mock in the EIE actually spares the gray matter, where on the contrary, with myelinolegodendrocyte glycoprotein associated disease, there is this characteristic H sign, which actually is specifically affecting the gray matter, and it's not quite clear yet how that actually happens. So we have been then interested more also in other like rare disease, another rare disease, to analyze these antigen-specific T cells more profoundly. And here you can see neuromyelitis optica that has been previously regarded also as a subtype of multiple sclerosis, but now actually has and has a female predominance, or 9:1, which is very clear, and also has a very similar clinic, like these longitudinal extensional lesions, as well as optic neuritis, to mock, and can be here distinguished by as well by a serum autoantibody against the water channel aquaporin 4, which you can see here, which is quite specific in these patients. You can just see the structure of the aquaporin 4 with several extracellular loops where the autoantibody binds. And here you can just see so the distribution of the aquaporin 4 is usually at spaces where there is water exchange, and these this protein actually builds like these rafts on the membrane that are called orthogonal areas of particle, where the antibody has been shown to preferentially bind to. And the general idea is that after the antibody reaches the astrocyte, it can induce both complement dependent and cellular dependent cytotoxicity to induce then the damage in the CNS. But then the question is, how actually can this autoantibody reach into the brain, as the aquaporin 4 is basically not visible through the blood-brain barrier. And our hypothesis was that T cells can facilitate the entry of those autoantibodies into the brain by opening the blood-brain barrier through inflammation. And here you can see an NMO patient's lesion with quite a lot of T cells compared actually to a MS patient's lesion. And here the OX40 staining with which is showing activated T cells also in neuromyelitis optica, proving that there actually is there are T cells involved in the pathogenesis. So we have also asked the question about the autoantibody pathogenicity, and here you can see as well, that at some point, if you inject the autoantibody alone, for some autoantibodies, nothing will happen, so no aquaporin 4 loss will occur in the lesions. But there are also autoantibody entities that are actually able to cause aquaporin 4 loss lesions. But what is true for all the antibodies is that if you have a T-cell induced inflammatory milieu, it always exacerbates the EIE and facilitates the entry of the autoantibody. And also those autoantibodies have been found and could be stimulated from patients, also with some molecular mimicry could be shown. And here you can see that although there are some preferential epitopes, you could say that in the different studies, the aquaporin 4 peptide is quite covered by the different specificities from the different species and also in one species. So what I've been looking at are aquaporin 4 specific T cells, also in a passive transfer model of EIE, to actually generate a model for neuromyelitis optica. And here you can see that we were able to reproduce the histology quite well with aquaporin 4 loss and IgG, as well as complement the position and infiltration of granulocytes into the lesions, and even could achieve with some cell lines inflammation in the optic nerve, although we could never see any aquaporin 4 loss. So still, yeah, it remains a model which could be further improved, perhaps. And when I went to Berlin, I wanted to ask the question, why the regulatory T cells are not keeping the balance in neuromyelitis optica patients, because it seems that there the disease is starting in the periphery and then going into the CNS. So it's not quite easy to find to define really the regulatory T cells, as they don't have a specific marker. So we have a marker combination, including CD25 high, CD127 low or negative, FOXP3 as a combination of markers for the regulatory T cells. And then we are basically reactivating the T cells with their specific antigen and looking at these activation markers. So CD137 being a marker for regulatory T cells, and 150 for effector T cells, also depending on the time dynamic, so that you can see here that the regulatory T cells are upregulating the CD137 before the effector cells would. So my two students are now have now established the protocol to isolate from our cohort the blood to perform the stimulation, and then we are going to analyze the cells with a spectral flow cytometer, and also have a single cell sequencing on these cells, and have a look at the methylation, as well as perform functional assays with the regulatory T cells to see if we can define what is actually the status of these cells in the NMO SD patients. And so here you can see some preliminary data where we could show that we are actually able to stimulate antigen-specific regulatory T cells in these patients. So this is here a 6-hour stimulation with overlapping peptides, and we could achieve like about a 30% activation in the regulatory T cells. And here you can see that without or with activation, you could have the impression, although this has still to be verified, of course, that the amount of regulatory T cells in an NMO patient is actually reduced compared to controls. Yeah, so we are now recruiting the patients and also analyzing, and hopefully soon, this year we will be finished with the data acquisition, and then I will be able to tell you more about the regulatory T cells and neuromyelitis optica patients. We have another open question, basically, because as you heard, like the aquaporin 4 is an astrocytic protein, right? Still, there's a massive demyelination going on also in NMOSD patients. So this is probably a topic that we will work on together with Becky to actually think about how is it that the pathology switches from the astrocyte actually to the neuron and myelin. Yeah, so that is our group that I thank a lot, our study nurses with the recruitment of the patients and my colleagues. And yeah, I'm happy to take your questions.
Thank you so much. That was terrific. Does anybody have any questions for Dr. Hastermann?
Hi, I have a question, Maria. Really nice talk there. So I guess that my question is about, do you think that if you do see that there's this change in regulatory T cells in NMO, do you think that also, number one, could have implications for MOGAD as well? And number two, do you think this is something that we could potentially specifically therapeutically target as a mechanism to target in people with NMO?
Yeah, thanks, Becky. So yeah, I mean, definitely. So we are also looking not only in NMOSD patient, but we also aim to have a look at the regulatory T cell compartment in myelin and MOGAD patients. Yes. So I think in general that perhaps some kind of maybe deficiency or lack or whatever in regulatory T cells is probably involved in many autoimmune diseases, right? It also has been shown in myasthenia gravis and colitis and Crohn. So there are several already diseases where it has been actually established that there's something going on with the regulatory T cell compartment. And I mean, basically our idea would have been to see if there would be like a cellular therapy that we could apply to the patient, where we say, okay, we can gain Tregs from the patient, expand them ex vivo, and then transfer them back and see if we actually could even achieve a curative effect in these patients. But yeah, that's maybe a longer term project.
Well, thank you so much for joining us today. This was really terrific. Your research is fantastic.
Thank you. I was very
Thank
happy
you.
to be here with you today.
Yeah. No, it was great having you. Bye, everyone.